So let's start. Hello, everybody. Thanks for being here today for a brand new webinar on biostimulant.com. My name is Alice Toderi, and I will host this webinar today. Before to start, I would like to give you a couple of pieces of information. This webinar is being recorded, and the recording will be available on the website biostimulant.com and on our social media in the next days. To interact with our guest, I invite you to use the Q&A box to submit the questions during the webinar, and then we will collect them and answer them at the end of the presentation. The topic of today's webinar is how biostimulants help tomatoes thrive under stress, insights from high-throughput phenotyping and metabolomics combined. And today, it's my pleasure to introduce today's speaker, who is Dr. Marzia Leporino. Marzia recently completed her PhD in plant and animal sciences at the University of Tuscia, specializing in innovative and sustainable horticultural production in protected environments. Her research focused on managing abiotic stress in tomato crops through the use of biostimulants to enhance stress tolerance, nutrient uptake, and fruit quality. She adopted an integrated approach combining advanced phenotyping technologies with omics sciences in order to gain deeper insight into plant responses under challenging conditions. So I think we can start. Marzia, the floor is yours, and you can go on with your presentation.
Okay, so good afternoon, everyone, and thanks for the introduction. And let's go straight to the introduction of this webinar. And let's see some data about climate challenge. We can see in the figure on the left how from the 19th century to today, the temperatures, the global temperatures always increased. In particular, we can observe a dramatic increase starting from the late 20th century, mainly depending on the greenhouse gas emissions and industrial activity. On the right side of this slide, we can see just the last of the 10 warmest years, starting from the 2015. In particular, we can see how dramatic is the situation during the last year, the 2024. This challenging situation relating the climate challenge, the climate situation all over the world, dramatically impact the agricultural production. In particular, we can see how the global vegetable production during the period from 2010 and 2023 increased steadily. And this production is significantly affected by growing demand for water, for irrigation water, for example, and abiotic stresses like soil salinity and heat stress that are increasingly, especially in the last years. Moving on to the tomato crops, in this figure we can see how widespread is tomato production all over the world. In particular, we can see the dark red color, which represent the most intensive production in most of the countries all over the world. Specifically, we can see how is increasing the tomato production in the world, mainly related to the increase in demand from the consumers and improvements in agricultural technologies, and also for increasingly intensive farming practices. When we zoom to the European Union tomato production, we can see how the trend is opposite, because the harvested areas in the European Union are decreasing. But interestingly, we can also observe how the production In smaller areas in the European Union, despite our decreasing, the production is increasing. However, we always need to take into consideration if this production is sustainable. Tomato, as one of the most consumed crop all over the world, is facing this agricultural and climatic challenge, in particular with a particular focus on the abiotic stresses, because we know that tomato crops are particularly sensitive to fluctuations in water availability, not only for the growth of the crops, but in particular for the flowering and the tomato yield production. As we can see that the drought, high salinity, alkalinity, as widespread abiotic stresses, especially from the Mediterranean areas, and also extreme temperatures can impair growth and nutrient uptake, leading to impaired carbon dioxide uptake by plants and photosynthesis, and also a reduction in the fruit set yield and fruit quality. One of the widespread abiotic stresses that are impacting in significantly the tomato cultivation is drought stress, because it represents one of the most frequent and damaging abiotic challenges for tomato growers, because water availability is essential for flowering and fruit development, and can cause, in the first place, stomatal closure, depending also from the extreme temperatures, for example, in protected environments, reducing carbon dioxide uptake and photosynthesis, wilting, leading to wilting plants, and impaired nutrient transport, which is fundamental and essential for the growth and fruit development. And as a final step, reducing the pollen viability, fruit set, and fruit development. If we consider that drought stress events can be repeated or prolonged during the crop cultivation, they can permanently stunt growth. Another challenging situation that can be present in crop cultivation of tomato is soil alkalinity, which can be combined often with drought stress, and especially in the Mediterranean area, can result from poor drainage, high concentration of alkaline ions, and low quality irrigation water, impairing tomato productivity by limiting the nutrient availability, especially when we consider that alkaline soils are characterized by pHs above 7.5 to 8.5, reducing the availability of key nutrients, for example, among macroelements, phosphorus, and among macronutrients, iron, zinc, manganese, boron, and copper. Alkalinity can cause, at the macroscopic level, chlorosis or yellowing of the leaves, can cause also stunted growth and reduced yield, and on top of that, can also hinder soil microbial activity, reducing the fertility of the soil. This is why often tomato growers use synthetic fertilizers that are able to chelate essential micronutrients to boost nutrient uptake. They are highly effective, however, they carry also risks of soil or water pollution, reducing the long-term sustainability of the tomato cultivation. So in this case, considering all the problems related to abiotic stresses, we need to look for sustainable solutions for plant resilience and quality. And we can consider products like and biostimulants like plant-derived protein hydrolysates. Why we can consider these products? Because they have a high plant compatibility, since they are derived from vegetal peptides and are easily recognized and absorbed by plants. They are effective at low doses, and in this way, tomato growers can reduce the external inputs, external input requirements, reducing the economic costs of the products and the crop in general, and also reducing the environmental footprint. Plant pHs, we can call in this way from now on, can stimulate, their action is multifunctional because they can stimulate growth, flowering, yield, fruit quality, and also nutrient uptake, enhancing also the plant tolerance to abiotic stresses like drought and alkalinity, for example. In this sense, they represent sustainable agricultural input because they serve as natural product and chelating agents. So they are eco-friendly, biodegradable, and non-toxic, and they minimize the environmental impact on the crop cultivation. Despite the use of this product, the pH and biostimulants in general have gained huge interest both by the research field and farm growers. Their mechanism of action is still unclear, especially at the molecular level. Despite the huge scientific literature that highlight the several positive effect that they exert on plants. Because they have multifactorial and complex responses on plants involving biochemical and morphological and physiological changes, they also represent a solution that it's highly context dependent. And in this way, they possess a limited predictability, since plant responses vary with environmental condition, timing, and application method. This is why we need for deeper understanding, because we need to optimize formulations and, for example, application methods, and also support the regulatory development and the confidence of farmers in using these type of products. This is why, as a solution to try to fasten this process, we need to use quick evaluation of pH's efficacy. We can evaluate the pH's efficacy conducting screening tests to assess the plant performances, particularly using targeted screening trials, evaluating the plant performance under defined condition, well-defined condition and controlled, using specifically short-term experiment that enables to study in a short time the response of plants simulating different abiotic stressing using different treatments. Identifying critical windows of action. In other words, we need to identify the specific plant developmental stage in which biostimulants and pHs are more effective, and monitor physiological, morphological also, and biochemical responses to track the plant adaptation under control conditions to different treatment and abiotic stresses. In this sense, we can combine innovative technologies to meet the need for a quick evaluation of pH's effect on plant tolerance against abiotic stresses, because we need to consider that traditional phenotyping is time-consuming and also despite valuable, it also offers a low scalability and a limited resolution for the results. In this case, we can introduce combined use of innovative and advanced technologies like 3D laser scanning, which enables high throughput and non-invasive analysis of the plant shape and health, and metabolomics analysis for detecting the metabolic shift under stress or treatment conditions. In the specific case, combining these technologies can give an opportunity to reach precision agriculture methods using biostimulants with purpose, timing, and reliable results. We can see here in the video a tool that can be used for the 3D imaging, which is used for quantifying both morphological and physiological changes with using 3D scans. This instrument used the pixels that correspond to the shape of the plants that are elaborated through the triangulation, generating point clouds that give as a result the real shape of the plant, to which are related various measurable traits, like the digital biomass, 3D leaf area, plant height, and different vegetation indices that are linked to the plant state of health. And this is called the sensor-to-plant approach, since it enables precise data collection over time in a non-destructive way. Combined with the 3D imaging, we can use metabolomics analysis to decode the plant's biochemical response to different treatments, biostimulant treatments, and abiotic stresses. We can use both targeted metabolomics if we want to explore the specific action effect of a pH on a specific metabolite that is produced by the plants, or untargeted metabolomics that is useful because it enables to study in a simultaneous way hundreds of metabolites. So in this way, we can identify stress biomarkers and specific metabolic pathways or metabolic shifts under different treatments, providing the answer to why we can see at the macroscopic level using the 3D imaging or the real biomass determination, why the plant changes in response to different biostimulants or different abiotic stresses set up by the experimental design. Here we can see now the experimental framework coming from my PhD research. In particular, we'll see two studies, and the first one was focused on how protein hydrolysates can enhance tomato plant resilience to multiple drought stress events. The experiment took place at the experimental farm at the University of Tuscia. Fresh tomatoes were grown in sandy loam soil, and the crop cycle went from May 20 to June 22. We used manual fertigation, applying a standard nutrient solution, and we applied two irrigation levels, one optimal and one suboptimal, in which the irrigation was stopped until the plants reached the wilting point, and then the irrigation was restored to start another drought stress cycle. Treatments were applied weekly, and they were an untreated control, emal-base protein hydrolysate, and FA-base protein hydrolysate. And the data were related to the phenotyping 3D laser scanning and the metabolomics analysis, the main results. As we can see here, we have the main results in relation to the digital biomass and the digital biomass traits recorded by the 3D laser scanner. And we can see the trend, the growth trend, both for the 3D leaf area. Sorry, I can maybe. Okay. The 3D leaf area and the plant height. And the product of the 3D leaf area and plant height is the digital biomass. We can see the black vertical line, which correspond at the days in which the treatments were applied. And we can see also the negative peaks corresponding to the days in which the wilting point was reached by all plants, and the following recovery period. In this case, we can observe how the pH1, the emal-base derived pH, was the best performing product compared to the untreated plants and the FA-base pH, which is the green line. And also how it improved the biomass development. In this case, we can also observe how the 3D leaf area was the main factor affecting the digital biomass. To validate this result, we also calculated the slope. In other words, the regression line between the wilting point and the highest peak at the end at the recovery period. And we observed how the pH1, the emal-base pH, was always higher during the recovery, effectively improving the recovery from drought stress in each water stress cycle. Here we can see 3 of the different vegetation indices that are related to the plant state of health. In particular, we see the NDVI, MPCI, and PSRI. NDVI is one of the most used indices to evaluate the plant state of health. And in this case, you can see 4 graphs, 4 trends corresponding to 4 bins that are the ranges in which the percentage of the pixels of each index is recognized by the scanner. In this case, for example, for the NDVI to -1 to until the 0.3 range, we can see the pixels recognized are at a very low percentage because all the plants were in a moderate good state of health. The MPCI is a chlorophyll or carotenoid ratio related index, and it's an index that decrease when the chlorophyll increase. And PSRI is an index related to mainly to the senescence of the plants. And in this case, because the plants were grown only for 1 month, it's a less reliable index in this case. As we can see here, the majority of the pixels of the NDVI fell within a moderate state or very healthy state of the plant. In particular, we can see how the pH1 was always was higher in comparison to the other treatments in the majority of the phenotyping days. And the situation was the same for the MPCI in which these ranges correspond to healthy plants. And the same for the PSRI, because it correlates highly with the MPCI for chlorophyll content. When we move forward to the metabolic profiling, we dive deep in the comparison between the best performing pH, the Malvasi pH, in comparison to the untreated plants. And we can see here the metabolites classes, the compounds that were significantly modulated by the pH in comparison to the untreated plants under stress situation. The red color corresponds to the metabolites that were increased by the pH. The blue color corresponds to the compounds that were decreased, and the purple one corresponds to the both increased and decreased metabolites in the pH treated plants. In particular, we saw that most of the metabolites that were increased by the pH were involved in all the processes that are triggered during the stress cycles. In particular, we saw an increase in fatty acids and sterols related to the transpiration processes during the stress cycle. A huge number of compounds that were also involved in the osmoregulation and aquaporins as unsaturated fatty acids. But the most interesting part for us was related to the dipeptides that we found as proteinogenic dipeptides that were involved in the gluconeogenesis as the as substitute of other compounds during the carbon flux during the water stress cycles, and they were also involved in the antioxidant activity. As we conclude, we can confirm how combining high-throughput phenotyping and metabolomics is highly effective to non-destructively at the beginning using high-throughput phenotyping. The effect of pHs, in particular pH 1, for promoting plant recovery after multiple water stress events. In this case, 3D imaging enables to confirm how the 3D leaf area was the main phenotypic marker, because it affected mainly the digital biomass trend during the crop cycle, and how metabolites such as dipeptides were involved in enhancing plant tolerance to abiotic stresses. And in this trial, we saw that the pH 1 can represent a promising protein hydrolysate for developing a functional biostimulant addressed to mitigate drought stress in tomato crop. Moving on, study 2, we also explored the potential of vegetal chelating peptides to support plant performance under alkaline conditions. In this case, the experiment took place at the experimental farm of the University of Tuscia. We used quartz sand to isolate the treatment effects to cultivate fresh tomato. And the crop cycle lasts always 1 month, more or less. We used automatic fertigation to supply an alkaline solution at a pH of 8.8. And we used to apply weekly treatments, comparing a biochelate, which composition was on vegetal peptides, chelating peptides, and the synthetic chelate. Both products were supplied at 3 different rates: B1, S1, B2, S2, B3, and S3, respectively low, medium, high rate, that were balanced. Each rate was balanced on the same supply of micronutrients. And because of the inert nature of the substrate, an untreated control in this case was not considered. In this case, we wanted to have a multi-layered data analysis, considering also the real biomass to correlate to the phenotyping data, the ionomics and total nitrogen to study the uptake, the actual uptake of nutrients by the plants, and the metabolomics analysis. Looking at the real biomass, we observed that there was a similar growth concerning both the fresh weight and the dry weight of the plants in all treatments. And especially we saw that the bioacrylate had a comparable effect to the synthetic fertilizer, even under alkaline conditions that were imposed by the experimental design. And even, as we can see in the photo, the 3 plants above are the bioacrylate treated plants, and the 3 plants on the downside of the photo are the synthetic clay fertilizer treated plants. And we can see how the chlorosis symptoms were more evident in the bioacrylate applied at a lower rate. However, this result did not significantly affect photosynthesis during the crop cycle. We also compared making a correlation matrix between the real biomass and the digital biomass traits. We observed that the real real biomass traits were strongly correlated to the digital biomass traits, confirming the reliability of this high-throughput phenotyping platform for the non-destructive monitoring of plant development during the crop cycle. Moving to the growth parameters, we can see 2 distinct phases in the digital biomass: an initial linear growth from day 1 of phenotyping to day 7, which potentially reflect the post-transplant adaptation. And from day 7 onward, we saw an exponential growth with no significant differences between treatments. And as we saw in the real biomass, we can confirm that the bioacrylate performed in a similar way to the synthetic fertilizer in stimulating early plant development. And also in this case, as we saw before for the drought stress experiment, we can see how the digital biomass has a strong relation with the 3D leaf area and the plant height. In this case, we did not evaluate the different vegetation indices related to the plant health, plant state of health, but we observed the average value for the NDVI, the chlorophyll carotenoid ratio, the senescent index, and adding also 2 other parameters, which are the green leaf index, which represent the greenness of the plants, and the UE, which represent the color of the plants. And it's expressed in a range from 0 degrees to 360 degrees. And each range corresponds to a different color. For example, for the green one is more or less from 75 degrees to 135 degrees. In this case, we can see how during the first phenotyping days, the NDVI increased because of the natural plant growth. and then stabilized, because probably in this case, despite the early development of chlorosis symptoms, the index saturated, and because it's mainly related to the biomass development, and it was not able to detect the yellowing of the leaves. In contrast with the MPCI and PSRI, which partially corresponded to the NDVI results, also always in the first phenotyping days. And both parameters increased during the crop cycle, indicating that they are more sensitive as indices of the plant state of health to the increasing yellowing of the leaves related to the chlorosis symptoms. In this case, the green leaf index was more related to the NDVI despite these fluctuations. And the same was for the UE, because the ranges always fell within a green range. Concerning leaf mineral composition, we can see here how the both synthetic chelate and the biochelate affected the macronutrient uptake. In both cases, fertilizer type and dose had no significant effect on the macronutrient uptake. In particular, we can see how nitrogen and calcium were under optimal ranges for tomato crop in greenhouse conditions, while phosphorus and potassium were below recommended levels. In the case of phosphorus, probably due to the high pH. As we saw before, soil alkalinity can significantly impair the phosphorus uptake. And in the case of potassium, there was probably competition within with the sodium uptake. Concerning macronutrients, the situation was more complex, since we can see here that iron was consistently below the optimal ranges for the tomato crop in greenhouse. And this was probably due to the reduced stability of both peptide chelate and the EDTA chelate complexes at high pHs. While we can see that manganese, zinc, copper, and boron showed all the dose-dependent responses. Most of these macronutrients were below optimal ranges, highlighting the need for the optimization of fertilizer dosage, both for the synthetic chelate and the biochelate. However, we can see, for example, how boron stay below optimal values in all treatments, while copper was deficient only in B2, but reached acceptable levels with higher rates of both biochelate and synthetic chelate fertilizers, while zinc seemed more improved in its uptake from the synthetic chelate fertilizer at higher doses. In the case of metabolomic profiling, we compared the same rates for the biochelate and the synthetic fertilizer. In this case, we can see, we can observe the results for the comparison between B1 and S1, which is the low rate of both fertilizers. And we can see how only two metabolite classes were significantly modulated by the biochelate at the lower rate. However, we observed that prenol lipids were mainly increased. For one example is methylgibberellin, highlighting that B1 probably promoted hormonal modulation for stress resilience under alkaline conditions. In B2S2 comparison, the situation changed, and we can see how the metabolites classes increased, the significantly modulated classes increased. And in this case, we identified a lot of compounds involved in antioxidant strategies, favoring mainly phenolic compounds, for example, for compounds related to the cell wall fortification, antioxidant activity. There were also monoterpenes and indole alkaloids, which are mainly related to pathogen defense. And last, oxyfolate pathways that are mainly involved in the growth, lipids, and chlorophyll and lignin biosynthesis. In the higher rate, in B3 versus S3 comparison, we also observed a shift in the phenylalanine reallocation for the lignin biosynthesis, which served as a cell, also in this case as a cell wall fortification during stress events, as the alkaline condition that were set up, and the mixed regulation of organoxygen compounds related to the stress tolerance of the plants. And also an increase, also in this case, in phenolic compounds related to the antioxidant activity during the stress. Concluding, we can say that biochelate was as effective as the synthetic chelate, both when observing the fresh, the fresh and dry biomass, but also as a reliable product that can be used, that can be studied non-destructively using non-invasive technology, as the 3D phenotyping platform, which is also a reliable instrument to study the effect of the products and under alkaline conditions, despite the fact that indices like NDVI were less reliable to detect changes in chlorophyll content in leaf tissues. Also, we observed how micronutrients for sure need to be revised in relation to the dose, to the rate. And we saw that EDTA seemed more stable for the copper and zinc uptake in comparison to the peptide biochelate. However, we interestingly observed how biochelate improved stress resilience by enhancing especially antioxidant defense, such as phenols, and hormone modulation also at lower rates. Considering that as a final step, biochelates, particularly at higher doses, can provide additional benefits of synthetic fertilizer because they are basically biostimulants, and they also possess of the biostimulant properties of vegetal peptides. As we take a step back to the overall results of both trials, we can confirm that the combination of advanced phenotyping and omics sciences such as metabolomics and genomics can help to reveal both the macroscopic effect of the biostimulant under different abiotic stress conditions, also helping to understand the mode of action of these products, identifying, in the case of metabolomics, specific metabolic pathways that are linked to the stress tolerance in plants and nutrient dynamics. We can say also that protein hydrolysates, under their double role of biostimulants and chelating agents, can promote both drought recovery through the metabolome modulation and exert positive action on the antioxidant activity and signaling. And acting as natural chelators, they represent a sustainable alternative to synthetic fertilizers. In relation specifically on the last experiment, we can surely optimize the application rates and, for example, also the stress intensity and the time of application of the products. We can also integrate other precision tool for early detection both of the stress and timing, the best timing for the application of the products. We can also think about exploring synergistic treatments to boost the efficacy of the products, and maybe evaluate the economic sustainability for the broader adoption by the tomato growers. Thank you for the attention, and I want to make a special thanks to my colleagues and supervisors, which supported me in my PhD journey. And if you have a question, I'm available.
Thank you very much, Marzia, for this insightful presentation, very rich and full of ideas for the future. I encourage everybody to submit the questions in the Q&A box or through the discussion. So I have already some questions for you. First one, thank you for the interesting presentation. Can you provide more information about the environmental benefits of using vegetal peptides, vegetal peptide-based chelates versus synthetic chelates?
Yes. The main positive outcome that we can gain with vegetal peptides is that they are biodegradable and eco-friendly, since they derive from vegetal matrix. In contrast to synthetic chelates that maybe on a short term they can be highly effective in providing micronutrients to the plants, but on a long-term sustainability point of view, they also can be toxic both for the plants if they are used in high quantities, and also they can be leached and for example remain in the soil, providing toxicity both for the plants and for example for soil microbials, and leaching also in the water causing water pollution, for example, having health risks both for humans and animals, while all these problems are not present as we use vegetal peptides because they are vegetal. Maybe one negative point of view could be that being biodegradable, they could be less effective and need more application. Yeah.
Yes. Okay. Sure. Anything else? Because we have a couple of other questions. So one is very articulate. It says, thanks, Marzia, for your clear presentation. Please allow me to ask the same questions about the concern I have. One, have you already worked with drought stress, or are you planning to apply it in your research? If yes, have you already applied or plan to apply microbiome-based strategies to induce drought tolerance in tomato plants? And what biochemical tests will you use to select the best microorganism to enhance drought stress tolerance?
Sorry, can you repeat the question? It's
So basically,
articulate.
if I understand well this question, it says if about drought stress, have you already tried to apply some microbiome-based biostimulants, or anyway, some microbials? To,
Okay.
the first one says to induce drought tolerance. So I suppose that it is to improve tolerance
The
to drought.
plant growth, maybe. Okay. No, the study that we saw, it was the first trial, the first experiment of my PhD journey, and then applied the drought stress, for example, on the long-term experiment that is now under submission, the experiment, the results under submission to a journal, a scientific journal. However, I never applied microbial biostimulants because at this moment I never considered that these products for my research, but it surely could be interesting to see the synergistic effect of microbials and pHs, for example, or only use microbials for improving drought stress, maybe because we know that, for example, mycorrhiza or trichoderma can exert positive effects, both as biocontrol and growth improvers for plant growth.
Okay. One question is very specific.
Mm-hmm.
In study 2, why did NDVI fail to detect chlorosis while NPCI and PSRI succeeded?
Okay, yes. The NDVI is mainly related to the biomass accumulation. So we saw before, maybe I can go back to the slide.
Yes.
Okay. We saw before that in the first days of phenotyping, the NDVI increased because the plants at the start of the trials of the experiment, the plants were small. Then the NDVI followed the natural increase in biomass in each treatment. However, it stabilized because being related to the biomass, the calculation, the mathematical formula that gives us the NDVI results, it's not able in this case to detect the changes, also small changes in chlorophyll content, while MPCI and PSRI being calculated on the pixels corresponding mainly to the yellow leaves, they are more sensitive in this way to the changes in chlorophyll content, and they are more sensitive in detecting and revealing the chlorosis symptoms on the leaves.
Okay, perfect. One question about this, are these biostimulants efficient in case of the presence of phytoparasitic nematodes attacking tomato? Do you have any study about that?
No, I don't have any study. They could be, I think they could be maybe effective because if they can enhance the plant resilience against abiotic stresses, maybe they can also, the nematodes can be able to attack the plant that is in a good state of health.
Sure. Thank you for your very clear explications. I have just one question about the 3D scanning. Can it be used by the farmers, or is it used only in laboratory?
It can be used both at a laboratory scale and also at the field scale, because for our specific case, we have a structure that enables the 3D scanner to scan the plants in protected environments. But there are other structures that enable the scanner to scan the plants at a field scale. However, you need always to consider the costs because they are really expensive tools, and you need to consider maybe at large companies can use, or in a society of companies.
Okay, so I would say that we have just the last question, and for you, and thanks again, Marzia, for your time. Can you expect more activity of vegetal peptides as foliar or root application?
I saw different works, different papers that highlight a better action of the protein hydrolysates as root drench because they are more easily absorbed. Maybe also considering also the timing of the application. Myself, I performed last year an experiment comparing root drench and foliar application. However, the drought stress that I imposed was too severe, and then the pH's effect was maxed in a sort point of view. However, in some cases, the pH applied at the roots was most effective.
Okay. Well, thank you, Marzia, for this session, and thank you everybody for attending the webinar today. If you still have questions, you can use the website biostimulants.com in the specific section, Ask the Expert, and we'll try to provide an answer. Also, I would like to remind you all that the recording will be available soon. Have a nice day, and I hope to see you again in the next webinar of biostimulants.com. Bye-bye.
Thank you. Bye.
Bye.